Nir.
Yes. So first of all, just conflicts. I'm showing you those two companies, not because I'm going to talk about them, I'm involved in them, but to show you that aging is beyond the labs now. They're biotechs, they're venture capital that are preparing us so that when pharmaceuticals will go into the biology of aging and stop aging, we'll be ready for them. That's the reason I'm doing that. What I decided to do now is to, yesterday, it's not me asking questions, lots of speakers said, Nir will talk about it. So now I have to talk about several things, not everything. But I want to make one point, and it's a very important point. What you see here is some subject in my study. I have 700 centenarians. Those are siblings that were born in New York City between 1910 and 1920. And each of them got to beyond the age of 102. When the young sister then on the right died at 102, they were all shocked. They continued to live to 110, 109, and 107. The woman on the left, Helen, was also smoking for almost 95 years. And when I asked her, none of your doctors told you to stop smoking? She said, all 4 doctors that told me to stop smoking, they died. Which makes the point that, first of all, if you smoke for 95 years, you live long life, but they were protected from the environment. And the most important thing, and I have 3,000 people in my studies because I have offspring of centenarians, I'll bring them in a second, but the most important question before we're talking about the science is, did they get sick when everybody gets sick, and then they lived sick for 30, 40 years, or were they healthy? And this is the answer, and it's 2 studies from Tom Pearl's in the New England Centenarians and ours. Maybe concentrate on the blue and green line, that's our study. And the green lines are our control, and the blue lines are our centenarians. And you see that their health span, as far as those diseases, their cancer, CVD, all written up, they were healthier for 30 years longer. Not only that, if you see at age 80, 10% of our control didn't have any disease. And after the age of 100, 30% of our centenarians didn't have any disease. And that brings me to the most important thing. It's not only their health span, but they had contraction of morbidity at the end. If we are sick for the last 5 years, 8 years of our life, they were sick for the last 5 to 8 months of their life. And studies like that and other have really led us to look at what we call now the longevity dividend. And I think that's the first most important point that I want to make. When we go to the politicians and say, we want the elderly to be healthy, I don't think that's the point. I think the point is, how much money are we going to solve by extending health span even by 2-3 years? And the answer is many trillions of dollars, more than any slide that you showed here of if we prevent heart disease, because the idea is that you prevent all diseases and you can prevent health costs. So I think when we come to the politician, we should talk about longevity dividend. Dana Goldman and Joel Shansky have actually an instrument, a tool that you can get to your states or to other countries, and you have to put just several figures, and you'll get the actual saving in cost. Okay, and I think that's very important for us to use because we cannot afford not to target aging. The second point I want to make is around biomarkers. And I'm much more optimistic of biomarkers. People were still talking about all biomarkers or methylation. The problem with methylation is it's not all reversible. We have to figure that out. But I want to give you an example that I think is going to be mainstream. It's in revision in Nature Medicine now. But the challenge for us is to distinguish between biological and chronological age, okay? That's why I'm taking centenarians, because their aging was really delayed. And we used this new aptamer technology by Sommermeyer, and we looked at 5,000 proteins in 1,000 people. You understand the amount of data and artificial intelligence that we're using now. And just that you know, when you get those data, you have to be prepared for those volcano plots. Okay, so we have lots of data in the volcano, and the volcano shoots up those red rocks, and the red rocks have to go high and also far away. The height is there significant. The p-values here are 10 to the minus 80, okay? And how far is how effective they are. And I'm not going to show you what they are, but to tell you that when we look at what they are, again, something that I doubt if you can read, some of them are mechanistic. For example, a lot of the IGF insulin signaling pathway is what we measure here. Okay, we knew it, we have genes for that, we knew that. But what's in red, it's all breakdown. It's degradation of collagen, of extracellular matrix, degranulation of white blood cells and other cells and thrombocytes. And at first, I was really disappointed. I said, I want to know the mechanisms here. But on the other hand, however we're going to treat aging, I think what we're going to monitor is the decrease in this breakdown. And why am I saying this is going to work? Because I didn't tell you the whole truth. 500 of those people were just regular people between 65 and 95, while the other 500 were the children of centenarians. And what you see in your eyes, the children of centenarians are more pink. They don't have that much significant. In fact, they lost 379 of the proteins because their aging is delayed. We showed that many, many times. So they're losing some of the proteins that goes up with aging. On the other hand, they have 29 proteins that the other people don't have. And what's interesting in those proteins is many of them are known as protective. For instance, the clotoprotein or the VCAM. So we have a biological measure for aging. We know this biological aging reflects biological age. We're creating the clock. I just want to mention another thing that's very important in aging. There's gender effect to everything we're doing. There's a sex effect to everything we're doing. The drugs are not, many of them are not doing the same in male and female. And what's interesting here, the proteome of females is much more stable. They have much less changes than male. And that's going to be interesting on its own. So the proteins that we thought are important, like IL-6, somebody mentioned it later. It doesn't even come as significant here. Okay, we have much better proteins that are coming up, and we're developing the biological clock. Okay, how are we translating what we showed yesterday? How are we translating to treatment? And our problem, unlike other opportunities, is that the FDA doesn't approve aging as a disease. And if that happens, healthcare providers are not going to pay for the treatment. And if the healthcare providers are not going to the treatment, the pharmaceuticals are really not working with us and the biotechs until they know there's a pathway forward, because the pharmaceutical need a business plan. Okay, so how do we get over that? So we used metformin as a tool, because we know that when we give metformin to animals, they live longer and healthier. And we know from clinical trial that metformin delays type 2 diabetes, that it delays cardiovascular disease, that there's association between those who are taking metformin and less cancers. There's several already clinical trial that shows that it prevents MCI and Alzheimer's. And I think this is really the interesting data here. And this is something that we should do in the United States. We cannot do it in the United States. This can be done only in England. Taking 200,000 people, basically, and looking at their death by the green is people with diabetes and metformin. And they had the lowest mortality over 5 years. They have lower mortality than people without diabetes that were totally matching them. Same pharmaceuticals, same doctors are treating them, everything. Okay, metformin had less mortality in diabetes, more obese, more sick people than control. And of course, much more than people who were treated with sulfonylurea, that is another drug to treat diabetes, but with diabetes you have increased mortality. So the point is, with all this data together, okay, we went to the FDA, okay, and we said, look, we want to repurpose a drug. We want to prove a concept that aging can be delayed. And basically, we sat with the FDA, and Thomas asked me to say, you know, we were sitting with the FDA about two things. One is, what's the indication? Okay, we didn't want to call aging a disease. We didn't want to do it. It's not necessary. The organization, ARP, wouldn't support it. Ageism is big. Elderly people are fired. They're not hired. Now you're calling them sick too. Okay, they don't want it. But we don't have to. We're going to prevent a cluster of age-related disease. To prevent, really, age-related disease, we do it, they don't care, by targeting the biology of aging. This is not an obstacle. But the question for them was, okay, we think of a cluster of disease that we can prevent. Is that okay? And they said, yeah, everything is okay except diabetes. Take diabetes out. I said, why can't we prevent diabetes? They said, well, look, diabetes is a chemical diagnosis. Okay, it's hemoglobin A1C. And if you watch it, those people who are diagnosed with diabetes, 10 years, 30% of them will have complications. That's not like having a heart attack, having cancer, having that. I mean, it's a total different way of looking at that. So we have a cluster of age-related disease that doesn't include diabetes. I'll stop here, and if there are more questions.
Okay, we're going to go on.